For Unit 6, we will be discussing DNA, RNA, and protein synthesis. DNA is our genetic material. It is what holds the entire code to make up who we are. And really, when it boils down to what that code is, it's really to make proteins. We've learned that proteins are essential for cells to grow and develop. It is what makes all of our enzymes, which do all of our functions in our cells, as well as give us our physical characteristics. For example, the protein melanin is what gives you your skin color. Keratin is in your hair and nails. Collagen is in your skin. So all of these things are helping us to become who we are. When we talk about the main concepts with molecular biology, which is about DNA, we call the central dogma the idea that DNA holds the code. RNA is then going to move the code to a place where it can be translated, and then that RNA is translated into a protein. Okay, so the DNA holds the information to make all the proteins in our body. The field of DNA science is relatively new. 1947, a man named Erwin Chargaff figured out the basic components of DNA. He figured out there were bases, A, T, C, and G, and he also figured out which ones paired with each other. In 52, Hershey and Chase figured out that DNA is genetic material. Before that, people thought that protein was the genetic material. So that was a big breakthrough. In the 50s, a woman named Rosalind Franklin used X-ray crystallography to get a picture of DNA and figured out it was a double helix. And then in 53, two men, Francis Crick and James Watson, built a model and then won the Nobel Prize for discovering DNA. Although it was a group effort, there was a lot of different people that contributed to that discovery. So DNA basically stands for deoxyribonucleic acid. It's made up of nucleotides. So if you remember back to Unit 2, we talked about nucleotides are the building blocks of nucleic acids. So we already know that those are the building blocks, or the monomers, but these monomers are made up of three smaller bits. So we have a deoxyribose sugar, which is the green one here. It's a central molecule. We have a phosphate group, which is the yellow, and then the nitrogenous base. And this is what differs for each nucleotide. These are always the same, a sugar and a phosphate. But then the one in the middle is going to be what makes up our genetic code. Okay, this nitrogenous base. The shape of DNA is a double helix, as you can see in this picture on the left. It has a sugar-phosphate backbone held together by covalent bonds. Covalent bonds are super strong and they hold together. So right here is a covalent bond between those two. So if you imagine a ladder, the sides of the ladder would be that sugar-phosphate backbone. And then the bases go into the middle and are held together by hydrogen bonds, which are very weak. So the bases would be like the rungs of the ladder going into the middle, and those are held together by weak bonds. There are four bases: adenine, guanine, cytosine, and thymine. They're grouped into categories: purines, which have this double ring structure, you can see here, purines, and pyrimidines, which have a single ring structure, those two right there. Adenine and thymine always bond together using a double bond. Okay, so notice there's just two bonds between A and T, and G and C always bond together using triple bonds. And it is because of this that causes your DNA to become a helix, because there's some double bonds, some triple bonds, and it causes the DNA to kink. If it were all just the same bonds all the way through, it would probably be a flat ladder. But because it's weird bonds every once in a while, it causes that double helix shape. RNA is a different molecule, but very similar. It's going to have a different sugar. It uses ribose sugar instead of deoxyribose. It has the bases A, G, and C, just like DNA, but instead of thymine, it has uracil. So we have a U instead of a T. And it's just a single helix, so it's actually a much smaller molecule. Okay, there's a couple different types of RNA. mRNA, which is going to be able to move, a messenger, so it goes from the nucleus to the cytoplasm. tRNA can actually take that RNA code and make it into amino acids. And if you remember, amino acids are the building blocks of proteins. Okay, so it's going to put together a bunch of amino acids. And rRNA is what a ribosome is made up of. So that is going to help to allow ribosomes to make protein. Okay, so some major things that are different between DNA and RNA. DNA is a double helix, has deoxyribose sugar, it is a larger molecule, it has the bases A, T, C, and G. RNA is a single helix, it uses ribose sugar, and it has the molecules A, U, C, and G. Okay, and that is the end.